narrow top. They both work just fine. You will find you have a personal preference with whichever one you prefer. I don't care which one it is. They both work. But if you have a preference, choose wisely. They've been in here now for about a half hour. I'm going to take my flask and put it on here for a moment. Turn that on. And I'm just very gently turning on the stir bar. I'm not going to have it on here very long, so I'm not going to mess around with heat. I literally am just getting it started up again to remix everything. But again, don't stir it so fast that you make bubbles or you'll be very sad because you do not want bubbles in your plates when you put them. So this is another connection. I have the other one on the other stir plate. Might as well just get that done now. This one though, since I'm going to leave it sit a little longer, the bead bath is set at 55 degrees. I'm setting this around that, close enough. It's fine. And it's still heating up. That's why the temperature showing as it is. But just let that stir so that when you're done pouring your first liter, your next one's going to be ready to go. So, take this, turn this off, and I'm heading to the hood. First thing I'm going to do, I am wearing gloves. I don't always wear gloves, but I think at this point, moving forward, everybody, let's plan on, we are wearing gloves. You have ethanol. For your gloves. Now your gloves are sterile. Just like your surface. There's another video on how to turn that on. Let them dry. When I worked in a research lab for a bunch of years, I worked doing tissue culture. I wore gloves, and this is exactly what we did. We would sterilize our gloves before we would work. So let's use that procedure here. I'm going to take my foil off. As long as I leave this in this space, I can just leave my cover off. But realize the way the air is flowing, I still have my covers on purpose because right now I'm reaching over them. If I had these covers off, I could not do this the way I am right now because you do not want to reach over anything. If I'm going to move stuff around, I will be doing it from the side. The way the airflow is, if I had anything on me, a fleck of a hair, any skin, any fuzzy, anything is going to blow into your work. Do not reach over anything, period. That's not optional. So, the way I am right now, I can get away with it because of what I'm doing. I am just going to set my foil cover aside. I take a couple of paper towels. This is what I always do. I just kind of fold it in half and I wrap it around. Why? Because it's going to drip. This way it's not going to drip everywhere. It will be fine. If it gets soggy, I can grab a new one. It's all good. I actually have 2 paper towels there that are just wrapped around. And now when I pour, notice my plates are nice and close to the edge so that the bottom of this has a place to be. If I'm pouring up in here, then I'm pouring too far and it's going to splash. Have it at the edge. And even this, I've changed my procedure over time. I'm doing one at a time. I am taking my cover off and I'm setting it face down out there. I'm setting it over here so I have enough room to do a few before I have to make another pile. I have one open. I'm pouring my one. And that's enough right there. You don't need to overfill it. If you have it half full, that's ample. You don't need to have it more than half full. I'm going to take my next cover and I'm not nesting it. I'm going to make a scaled back pile here. You do not want to nest them because I have now touched the outside. I don't want the next one that I put on there to end up being having the inside of the next cover touching the outside of this one because again the inside of each of these is sterile but the outsides are not. So you'll get a good rhythm going here where you can just do one. If it doesn't quite fill, just give it a little swirl, it'll fill. Do the next one. And now, if I was to reach back to that pile, I'm kind of reaching over things. So I'm just going to make a new pile over here. my row here, and then I'll talk through the next step. Pile. And I'm realizing I did not leave myself enough room to actually work with these last couple, so I'm stopping at this one. And I'm putting my flask down. See, if I would have poured these, I'd be reaching over them. So I'm going to leave those for next time, for my next batch. I'm going to go back to the beginning because these are starting to solidify. And I'm going to just, from the edge, push them to the back without, hopefully without stirring them around too much. And you're seeing I am not reaching over them at all. I'm only touching it from the side. And I'm also, one thing that I'm doing that you will not do while you're doing this, I'm talking. Talking is not a good idea. If you're doing this, you got to keep your mouth closed because I might be accidentally spitting in these as I am pouring. So don't talk. If somebody's pouring, you don't need to be talking over their shoulder. You also should not be walking behind here as somebody else is pouring because you're just creating an air current into the space that is not helping. So if somebody's pouring, leave them alone and stay away from the area. Now that said, if you're learning how to do this, stand nearby and watch somebody, that's fine. If you have a question, realistically, wait till they're done with the section, step back, talk. You really don't want to be talking over your plates because you're asking for trouble. These are to grow bacteria. We don't have bacteria in our mouth, right? So if I'm breathing on these, if I'm talking over them, they could be contaminated at this point. But this is a demo, so what we end up doing, this is why we take plates out the night before and leave them on the bench with the whole day ahead of a class eating them, because just in case there's any contamination, it will have grown overnight like that. So it'll show up and then we can make sure that what we're giving them is actually sterile. So I'm going to take my next batch, set them up. You want, as you're doing this, you want to work kind of swiftly because they are going to solidify. Even in here, it's agar. As it cools, it's going to solidify. It's pretty hot yet right now, so I'm not worried just yet. But once you get down near the bottom, don't leave it sitting here very long or it's going to be a chunk and you're not going to be able to use it. Not the end of the world. Just need to be aware that that's how this works. Now, as you're setting things out, actually, as I set out my first batch, I fumbled a couple, and I had one that flipped open, and when I caught it, my finger touched the inside of the cover. Guess where that one ended up? In the garbage. I touched the inside. You can't touch the inside of this stuff or you are contaminating it. So as you're doing this, now I can go ahead and wrap this around and keep using it and go ahead and pour my next batch, move them back. You can keep pouring until you fill the hood. Once the hood's filled, you gotta wait until whatever your last ones were at the front here until they solidify. Once they're solidified, so you can't reach over, right? Once they're all solidified, you'll be able to grab the covers. And I always just say, kind of sneak up on your plates to put the cover on. I don't want to grab and put them over the top because that's not keeping things sterile. You just need to literally sneak up on it and get that cover on so that you are keeping them as optimally sterile as possible. People do a lot of these pouring like this on an open bench. We're blessed with having such a nice clean space. This is a clean hood, that's what it's called, a horizontal clean bench, so that the space stays cleaner for us and it makes it that much better for you guys to work in. But it's still open and still has potential for contamination. So our goal is to give the classes the best that we can. You can see these are steaming up because these still are not solid. So I'm going to go ahead and take the covers back off just for the moment here. But again, you're going to start with the front ones. You have to wait for all of them to solidify because you're going to start at the front and work your way to the back. And then you can start stacking them. And remember, when you put them back in, when they're all covered, go ahead and put them into a stack in the direction they are. got your bag, you gotta finish filling out your bag. Who bagged it? Who poured it? Finish filling this out. And then with them stacked with the bottom side to the bottom, put them in a stack of 25 tall, slide this over the top, and then carefully flip them. And then they're in the correct position, because when they go in the fridge, the agar side is on top. The empty cover is what's at the bottom. That is 100% because of condensation. Because as they're in the fridge, they may develop condensation again. If you ever go in there and look after something has been in there for a few days, you'll see condensation. So you need the agar to be on top. So if your agar's in the small bottom part, then you got your cover. When it goes in the fridge, it's upside down like this. Same thing when you're growing things in the incubator. That's how you do it. You go ahead and spread your bacteria in there, and then it grows in the incubator like this. Feels weird, but that's how it works. So I figure the more you know, the more valuable you can be as a process here. You know why you're doing certain steps. If at any point you have a question, come ask. Even though you're seeing the video, you still may have questions. I might have missed something. That's fine. Come and talk to me. I'm always here to be able to answer questions. I will never be upset about anybody asking a question. You guys know that by now. So that's it for this then, because we got finished pouring our bits.